Yes, hello, my name is Sreena, and we are starting the course Animal Diversity here. The whole animal kingdom has been classified into two parts, that is non-chordate and chordates. So we are only studying the non-chordates part first. Under non-chordates, जो हमने first phylum पढ़ना है, that is Porifera, then Cnidaria, which you also call Coelenterella, and then third one Ctenophora, fourth one Platyhelminthes, fifth one Ascomynthus, sixth one Annelida, seventh one Arthropoda, eighth one Mollusca, ninth one Echinodermata, and tenth one Hemichordata. Okay, so under non-chordates, we are going to cover these 10 phyla. So let's just discuss all of them first, and then we will move on to the chordate part. So phylum Porifera. It comes under subkingdom Parazoa, so the study of phylum Porifera is also called parazoology. The term Porifera was coined by Robert Grant. Okay, so first thing first, the general characteristics of phylum Porifera, they are very simple animals. Okay, so they have pores in their body, everyone knows this, and they are simplest and diploplastic animal, and they have a cell aggregated body plan. That means they don't make up a tissue or organ as such, but they have function on a cellular level only, and all of them are marine. That means they love seawater, except we have an exception here. We have an exceptional animal here, which is Spongilla. Spongilla are the only poriferans who love fresh water. Okay, so before diving into the general characteristics, let me just make a rough diagram of what a sponge really looks like. Okay, this is a rough diagram of what a sponge would look like. Generally, it is vase-like structure. Then what you are doing is basically you are making a slit in between so that they become porous. Okay, so these are the pores here. All the pores, these are the small pores, okay, which is present in their body. These are called ostia. This ostia is actually the opening pore. This is actually the water inlet pore. From here, water enters to the sponge body, and this one larger one is the osculum. From here, water moves out of moves out of the sponge body. Basically, it is water outlet. The external body wall is called pinacoderm, and the inner body wall, they have this flagellated cell, which we call collar cell or choanocytes. So this inner body wall is called choanoderm. In between this pinacoderm and choanoderm, this one is called mesoglia or mesenchyme. Here you have all the cells which perform the digestive, nutritive, or reproductive, you name it, and these cells here do those functions. Actually, most of the cells here are amoeboid cells only, so that they can move here and there in the mesoglea, which is jelly layer. So the strength to the sponge body wall is given by spicules, which could be made up of calcium carbonate. So they are called calcareous spicules. Could be made up of spongin fibers also. Let me just draw the body wall here, body wall of sponge. So you have this pinacocytes, which is making the outer body wall, and the whole external body wall is called pinacoderm. Pores are made out of this porocyte. Okay, so this is the pore, which is actually your ostia, jelly, that jelly material in between the pinacoderm and the choanoderm. You have this mesoglea, and inside the mesoglea you have a variety of amoeboid cells. Then you have spicules, calcareous spicules, spongin fiber. You might have the siliceous spicules to strengthen, to provide strength to the body wall of sponges. And then the inner wall, inner body wall are having these, flagellated choanocytes. The layer is choanoderm. Okay, so this is basically your body wall of sponge. And then you have this choanocyte. This choanocyte is having a circular cellular body inside which there is a nucleus, and this is your nucleus. And then there is a parabasal body, out of which a thread-like rhizoblast will arise, and just at the periphery of this cell cytoplasm we have a blepharoblast, out of which the flagella arises. And this blepharoblast and flagella, these both are covered with the help of this collar. This is the collar, and inside this collar we have many microvilli. These are the microvilli, and these microvilli are actually adhered to each other and supported with the help of these microfibrils. So this is a sponge body basically. Here you have porocytes. Just assume here we have porocytes. These are the ostia. So the water will enter through ostia into the spongocoel. This one, the hollow cavity here is called spongocoel. So the water will enter into the spongocoel, and after entering the spongocoel, you have the choanocytes here. The regular beating of the choanocytes will pull the water inside the collar, and whatever food material, the food particle, the small particle it will bring in, it will adhere to the collar. Inside the collar you have mucus present there. So all the small food particles, they are going to get attached to this mucus, and this mucus will slowly draw this particle to the end of the cell. Inside the cell, these food particles are going to break down, and the thesocyte, There is an amebocyte, modified amebocyte here called thesocyte, which actually stores the food. Then after that, it will pull out the water from the collar, and that water will move out from this opening called osculum. Let us just discuss about the amebocyte. So these amebocytes, they are present in the mesoglia, the jelly layer between the pinacoderm and the choanoderm. So these amebocytes are going to get modified into many different type of cells. So the basic cells which have the capability to produce these different types of amebocyte, they are called archaeocyte. These archaeocytes are totipotent cells present in the mesoglia. They have the ability to generate, to give rise to different types of cell. They have the mitotic ability. Then we have chromocyte. Chromocytes are pigmented cell which imparts the color to the sponge. And then you have trophocyte. Actually, here this thesocyte could be trophocyte as well. Okay, so trophocytes are actually they also break down the food and give energy to the sponge. Then we have thesocyte. It stores the food particle. And then we have colonocyte, which secrete the collagen fiber to give strength to the sponge body. Then we have sclerocyte. They produce the spicules, which also provide strength to the sponge body. And then we also have phagocyte. They destroy the damaged cell. And now the reproduction. So reproduction could be both asexual and sexual. In the asexual part, majorly it happens through budding, and if they make internal buds, they are called gamules, which actually is a very specific character of your spongula. So this is what it looks like. The gamules, they have a lot of archaeocytes inside the gamule body. These are archaeocytes. Remember what archaeocytes were in mesoglia? They are totipotent amoeboid cell of mesoglia. Okay, so these archaeocytes are being protected by amphidisc spicules. Amphidisc means they have tapering end. Okay, so these kind of spicules, these are protecting these archaeocytes from outside. These are amphidisc spicules. Outer membrane. This is the outer membrane. This one is the inner membrane. Okay, and then in between this membrane you have one outlet. Okay, this outlet is called micropyle. From here water can enter into this gamule. This is micropyle. From here water can enter into this gamule, and the totipotent cell, this archaeocyte, will start dividing. Outer covering will break off and sponge will grow. So this is basically what happens when, in case of asexual reproduction in spongula, when budding occurs, these gamules are formed. And when we talk of sexual reproduction, there is always indirect development. That means They have larval stages in their life cycle. So they have two types of larvae in sponges. The first one is free swimming solid larva, and the second one is hollow larva, half flagellated and half non-flagellated. This one is called parenchyma, and this one is called amphilostula. These are the two types of larvae that are present in the sponge. Okay, so the Porifera has been classified on the basis of endoskeleton, majorly and minorly the canal system. Okay, on the basis of endoskeleton, we have three classes. Okay, so we have phylum Porifera here. So on the basis of endoskeleton, we have three classes. First one is calcarea. Second one is demospongia. Third one is hexactinellida. Okay, so the spicules here, they are going to be made up of calcium carbonate, and in this case they will be made from spongin fiber. And here the spicules are going to be 8 rayed. Okay, like this 1, 2, 3, 4, 5, 6, 7, 8. So these kind of spicules will be present here. Okay, so this is on the basis of spicules, and then on the basis of canal system, they have ascone type of canal system majorly, and there could be present cycone and leucone as well. And in case of demospongia, we are having this leucone type canal system, and which could be aphodal or diplodal. Okay, in case of hexactinellida, we have leucone type only. Okay, so the example of the sponges under class Calcarea are Leucosolania. First one is Kypha, and the second one is Leucosolania. And under Demospongia we have Euspongia and Spongilla. Under Hexactinellida we have Euplectella and Hyalonema. Okay, so this was the classification, and in the next video lecture we will discuss the canal system of sponges.